it is a twinning program between these two universities, the Babylon one from Iraq, and the second, which is Northampton University from the UK. So, and these people with me, all of them, except Eman from the University of Northampton. My work is developing a multidimensional fuzzy deep learning for cancer classification using gene expression data. I will start with agenda, introduction, challenges, the proposed model, experimental results, conclusion, and future direction. In introduction, cancer is defined as a group of disease or abnormal cells that arise in a specific area, then spread to other parts speedily. This is why cancer is very dangerous, and most researchers trying to improve their models for earlier detection. And starting these people with using CT scan, MIR, and etc. for cancer classification, which is provide image data set. Sorry, I hear some voice. Yeah. Okay. Then people trying to use alternative methods for providing some data sets, which is gene expression data, when it compared with a previous one was less cost, and as well as providing earlier detection of cancer by providing remarkable genes that could be used identifiers for training models, and then using these genes, whether this person has a cancer or not. And this help in earlier detection as well as in personalized treatment. Usually we have some cancers, especially, for example, breast cancer, and maybe this person has breast cancer, take different treatment based on which gene has activate in this person. So people provide different methods, and the most popular ones, microarray and RNA-seq. These tools, provides a gene expression data for between cancerous and non-cancerous samples. And these tools open the door for researchers for developing different mathematical and statistical methods for analyzing these huge data. One of these methods that have been used to analyze gene expression data was machine learning and deep learning. So machine learning is different from deep learning, and as we know, artificial intelligence is the big one, and therefore then machine learning is inside artificial intelligence as a topic, and deep learning inside the machine learning as a topic. The difference between machine learning and deep learning are the feature extraction. In deep learning, it will be inside, but whether by contrast, machine learning is independent feature selection we're using. Both are trying to, when I saying machine learning, that's mean deep learning included, because as I mentioned, deep learning is a subfield of machine learning, and machine learning is a subfield of artificial intelligence as a topic. Cancer classification, as I mentioned previously, researchers use different data sets, and one of them gene expression data, and previously using CT scan, MIR, and I mentioned there are some advantages with these data. For example, these data are expensive, as well as some people are scaring from taking the image inside CT scan, radiation in the data set, affect especially for children in the future. So this is why the researcher move on to other data set. Challenges with this data set, I mean, in gene expression data set, the high dimensionality, which is the number of features is greater than the number of samples. This is one of the most challenge in gene expression data. And this is why some people using different feature selection. Hi, you hear me? Sorry, I think I lost the. Do you hear me?
Yeah, now it's audible. Yeah, you can go ahead.
Oh, yeah.
Yeah.
Yeah. So this is the challenge in gene expression data set, which is high dimensionality, as well as in deep learning are often considered black box, which means we don't know which features has been selected for their decision. And the understanding the features of genes interaction contribute the model predictions is crucial for gaining insight into underlying biology. As I mentioned in the introduction section that maybe we have a cancer type, for example, lung cancer, and between two different people there's different treatment. So based on which genes are activated in this cancer. Additionally, we have many cancer data sets have limited samples, as this is one of the issue. We have number of samples is very small compared with other data sets, but features is very high. And this is very challenging to train a machine learning for taking their decision, and mostly related to a small sample size, such as overfitting, an example. This is one of the issue. As well as impulse data set, I mean, is, for example, we have in breast cancer, 80% as samples of the samples are cancer and 20 of the samples are normal. So in this case, imbalance data, this is one of the issue. Five challenge, the five one is model trained on one cancer type may not generalize well to other due to clinical differences. So and because the differences between each data set, even gene expression data for different type of cancer, for example, a model get an example 90% as an accuracy with a cancer type, and with other type maybe get difference. So this is another challenge to make a model very generalized to other for different data sets. So here we propose a model that starting from end-to-end process, which is starting with pre-processing. The data set include missing data set, including duplicate data set, and we normalize the data sets in different layers. Therefore, we applying fuzzy gene selection wrapper plus, which is already published in other work. It is, to be honest, with two concepts. Fuzzy gene selection, therefore, we using integrated with wrapper feature selection. So usually we have 4 types of feature selection techniques, and we call it here in gene selection techniques. We have filter, we have wrapper, we have embedded, and we have hybrid. So we integrating filter and wrapper methods in fuzzy gene selection wrapper plus method. After that, we make fuzzy classifier method, which is starting with 3 classifier models. Therefore, we apply fuzzification and defuzzification to propose. This another paper has been already published in IEEE Access journal, which is fuzzy classifier method. So in this model, we have using 3 feature selection, and we integrating these 3 feature selection, then we employing integration methods and Bayesian and Bayesian methods for getting a test score for each gene that will be selected as a biomarker genes, or that would be used as identifier for model. Once we have selecting from thousands of genes, as mentioned previously, high dimensionality data set, which means we have thousands of features and we have hundreds of samples. So not all these features are relevant. So this is why we're using fuzzy gene selection wrapper plus method to reduce the dimensionality and selecting top genes that highly correlated with the target or the disease. Once we select these genes, we move out to the classifier to train this model, and we using cross-validation method to make the model reliable and reasonable. The results that we obtained from this model. So when we here apply 6 data set to evaluate our model, and we compare them with existing classifier methods. For example, in our previous work, we mentioned that before and after we applied Bayesian gene selection wrapper plus method. So this is like complete from the previous publication. So here we compare 2 popular classifiers in this field using 6 gene expression, and we compare it against our model, and our model show it's better than these classifiers. For example, here, after we applying Bayesian gene selection on this dataset ID, we find out that our model achieved with high accuracy and precision recall and F-score, as you can see in the table in front of you. So we see in all these 6 data sets, our model achieved better than these classifiers. Here we compare our work with other published work using the same data set that have been used by these studies. For example, in a first study, passed with 88% as an accuracy, 82 as a precision, 97 recall, and 89 F-score. While our model only with 18 genes passed 93. So our model here demonstrate that used less number of features, as well as higher accuracy, precision recall, and F-score. As well as in the second study, this study doesn't mention how many number of genes that been used as identifiers for training their model. However, our model achieved higher accuracy as well. The other study is with the third data set, selecting only 90 features or genes, while our model chose 5 genes and achieved higher accuracy. So in total, our model achieved better accuracy and less number of features for training the model. Conclusion and future directions. The study developed end-to-end process, which is for the selection of informative genes and the precise classification of various cancer types using gene expression data set. So our model using 6 data sets for evaluating and testing the model, as well as achieving outstanding outcomes in terms of gene subset size, replication, and cancer classification accuracy, using a cross-validation method to reduce the dimensionality, to reduce the overfitting issue, and provide reliable results. Our model also in the also our method has our method also, sorry.
Yeah. Thank you, Mohammed. I would like to know if any questions from forum.
Sorry, I didn't complete the future direction because
okay, fine. Yeah.
Sorry
you
for
can
that.
Go ahead. Yeah.
So also our method is using different evaluation metrics to evaluate the model. So in total, our method reduced the dimensionality with a novel Bayesian selection wrapper plus method, and with Bayesian classifier for achieving the best accuracy, and generalize the method to classify different data sets, as you see in our tables, that using 6 data set, and our model show the high percentage in all these data sets. After that, so in future direction, the significant achievements, it is crucial to recognize the internet limitations of the study. So in the future, we can using different tools like RNA-seq. As I mentioned previously, we have dataset microarray and RNA-seq tools. So both they provide gene expression data. So our future, we will focus more on the RNA-seq, as well as we can use other different algorithms to compare our model. Additionally, investigate, however, so additionally, we can using some biologist techniques like enrichment, an example, to see how affected these selected genes biologically. And this maybe make our study integration between biologist and computer science view, rather than focusing on computer science view. So feature selection is really important when we're using these genes as for earlier detection. So next, in future direction, we will focus on this really important field. Thank you so much. If you have any question, I am more than happy